Hello, everyone, and welcome to today's webinar. I'm Melissa Romero, Forensic Science Consultant and CNEN Media Group. I will be moderating today's event, From Discovery to Cure, Addressing the Challenges of Oncology Drug Discovery. This educational webinar is sponsored by Eurofins Discovery. Eurofins Discovery built its industry-leading reputation on successfully delivering best-in-class drug discovery services to any organization, large or small. No matter where you are in the preclinical process, or if you need full end-to-end solutions, their experts support you every step of the way with purpose, precision, and innovation. As a global leader in drug discovery for over 40 years, they have strategically amassed a deep and broad range of over 12,000 assays for every type of research, as well as a full team of industry experts across disciplines to collaborate with you. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the top right corner of the screen or type your query into the Q&A box. If you're disconnected during the webcast at any time, please log in again according to the instructions that you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. This webcast allows a certification of participation. To receive your certificate, please click on the certificate icon below your Q&A box to download your copy after watching the webinar. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and each webinar will be archived at CNEN Online after the live webcast. During the presentation, we will be hearing from Michele Modugno and Dr. Alistair King. Dr. Alistair King is Head of Biology at Eurofins Discovery. Alistair has over 25 years of experience with all stages of drug discovery, from target validation to IND package generation, largely for oncology and inflammation. He has experience leading international strategic initiatives and drug discovery teams, for example, that which discovered Tafinlar, and the coordination of due diligence studies on external assets for in-licensing and subsequent clinical development, example for Mekinist. Michele Modugno is Scientific Project Leader in Biology at Eurofins Discovery. He has over 20 years of experience in preclinical oncology drug discovery. Michele has led multiple drug discovery programs, overseen project teams, and driven progress from target validation through to IND-enabling studies. He has also contributed to drug discovery for Danusertib and Encorfinib, trade name Braftovi. And let's get started. I will now turn it over to Michele.
Okay, thanks for your introduction. And first of all, I would like to thank the American Chemical Society for hosting this webinar and to thank you all for joining. So let's take a look at what's on the agenda for this webinar. We'll start talking about what are some of the key challenges in oncology drug discovery and how these challenges can be addressed. We will take a look at how undruggable targets are being prosecuted. What are the technologies and modalities that are changing the cancer drug discovery space? What can we do to minimize the risk of clinical failures due to unforeseen adverse events? And why it is important to understand as much as possible the molecule's mode of action before entering clinical trials. As we know, the drug discovery process is a long, multi-stage process that can be roughly divided in two big parts, right? Preclinical drug discovery and development and clinical experimentation. And here at Eurofins Discovery, we have all the expertise and capabilities to support preclinical projects, starting from target identification and validation to hit identification, up to clinical candidate selection and IND-enabling studies. We all know that major advancements have been made in understanding cancer biology, and that these have translated into novel and improved treatments. However, cancer still remains a very high unmet medical need, with high demand of new treatment approaches and therapies. Unfortunately, cancer drugs suffer from one of the highest attrition rates. As you can see from the graph on the left-hand side of the slide, cancer drugs have a likelihood of approval from phase 1 of only 5.3%, meaning that almost 95% of molecules that enter phase 1 ultimately fail during clinical development. And as you can see from the diagram on the right-hand side of the slide, the vast majority of these failures happen in phase 1, around 50%, and phase 2, around 70 to 80% of drugs that enter phase 2 fail in this phase. And this suggests that the factors that ultimately lead to clinical failures are either safety, which is the endpoint of phase 1, or efficacy, that is the endpoint of phase 2 clinical trials. And this is in agreement with what has been repeatedly reported by multiple analyses during the years, that a lack of efficacy and safety issues are the cause of 60 to 80% of drug clinical failures. So the question is, how can we tackle this problem? What can be done at the preclinical level to select candidates that have reduced probability of costly clinical failures? We obviously need to discover and develop drugs that have increased chances of clinical efficacy while minimizing chances of toxicity. But how can we do this? In this slide, I reported some of the key potential approaches. From an efficacy point of view, we know that targeting cancer driver genes has demonstrated to be a very successful approach, as exemplified by the many approved therapies that specifically target oncogene addiction mechanisms. Therefore, identifying novel druggable cancer driver is one of potential approach. But an additional approach is to develop novel ways to inhibit targets that are very well known to be cancer drivers, but that were previously considered undruggable. There is definitely a need to improve the in vitro and in vivo models that are used in preclinical drug discovery to test the drugs in models that are more translationally relevant. And having a better, more thorough understanding of what is the molecule's mechanism of action, how exactly the molecules work, can also help in, for example, identifying the right tumor context, the patient population with the highest chance of response, the best combination. Stratifying the patients using a biomarker of response is also demonstrated to have a very positive impact on the likelihood of approval, stressing the importance of identifying these biomarkers as early as possible in the preclinical drug discovery process. And from a safety point of view, profiling drugs on large panel of protein targets can allow to understand if the toxicity observed, for example, in preclinical models is on or off target, therefore understanding if the toxicity may be designed out by medicinal chemistry efforts or not, as well as profiling molecules on markers that are really predictive of toxicity. And finally, and this resounds with the previous comments on better understanding the mechanism of action, having a better understanding of the drug activities in biologically complex systems can allow to predict potential toxicities that would not emerge in the classical preclinical models. In the recent years, we have observed some new trends in oncology. Obviously, precision medicine, the possibility to tailor treatments to individual patients based on their genetic and tumor characteristics, has become more and more important. The exploitation of the immune system, as demonstrated by the success of checkpoint inhibitors. Novel modalities like CAR-T and antibody drug conjugates have obtained really significant clinical success. Small molecules still remain a key modality, with new interesting approaches like degraders and molecular glues that are emerging and are entering clinical development. I was mentioning the identification of novel cancer driver targets. One example of a novel target is the Werner helicase. Werner is a member of the RECQ family of helicases that is involved in the maintenance of genome integrity. And Werner was identified some years ago as essential for the survival of cancer cells that are mismatch repair deficient. These cells rely on Werner to repair DNA damage. Therefore, antagonizing Werner activity in these cells leads to DNA damage that is not repaired, and this ultimately leads to cell cycle arrest and apoptosis, while cells that are mismatch repair proficient are spared. And this kind of synthetic lethality rationale is very appealing because it allows, on one hand, to select the tumors that are more likely to respond, while on the other hand, to spare normal cells, resulting in a good therapeutic window. And as you can imagine, Werner inhibitors represent a promising therapeutic approach for these cancers. And actually, there are at least 2 molecules that have already reached clinical development, with several others in preclinical development. Since Werner is becoming more and more interesting in the oncology drug discovery space, we have developed several in vitro assays to test small molecule activity on this protein. Specifically, we have developed assays to measure both the ATPase as well as the DNA unwinding activities. And you can see on the left-hand side of the slide the 2 clinical compounds profiled on these 2 assays, as well as assays to measure direct binding of molecules to Werner using biophysics methods. But in a drug discovery program, it's very important to be able also to profile compounds for selectivity on close homologs. Therefore, we have developed and are still developing a suite of assays on a panel of helicases. And you can see them in the table on the right-hand side of the slide. And some of these are actually interesting targets themselves, like POLQ or DHX9, for example. And also for these proteins, we have ATPase, DNA unwinding, and biophysics assays. And we are also developing cellular target engagement assays. With these assays, we can now profile not only Werner inhibitors, but also inhibitors of other helicases in a very complete way. The best example of a target that was previously considered undruggable is KRAS. And I don't need to talk about the importance of mutant KRAS as a driver in many cancers, especially pancreatic and lung cancers. But it was only in 2013 when Shokat's lab discovered the switch to pocket that this oncogene became druggable. And since then, a lot of progress has been made with 2 drugs targeting the G12C mutation, sotorasib and adagrasib, currently approved. And I think at least 10 others in clinical development and many more in preclinical development. And this drug includes also allele-specific inhibitors targeting also other mutants further than the G12C, as well as pan-KRAS inhibitors. We recently published a paper reporting the development of a suite of KRAS assays for both wild-type and mutant, including several clinically relevant mutants, that measure multiple parameters, from direct binding assay to biochemical in vitro assays for measuring nucleotide exchange, to cellular target engagement assays, and even assays to measure KRAS degradation for KRAS degraders profiling. On the right-hand side of the slide, there is reported an example, a cellular target engagement assay that was used to test 3 inhibitors, 2 G12C inhibitors, sotorasib and adagrasib, and the G12D inhibitor, MRTX1133. And the results nicely show that in a G12C mutated cell line, panel B, we can appreciate target engagement for sotorasib and adagrasib, but not for MRTX1133 as expected. While in a G12D background, in panel C, only MRTX1133 show target engagement. And finally, in a KRAS wild-type cell line, none of the inhibitors is interacting with the target. And with this panel of assays, we are now able to evaluate in parallel multiple parameters that are relevant for KRAS inhibitor discovery and optimization. I will never underestimate the importance of structure-based drug discovery. We have recently obtained the crystal structure of KRAS G12C and G12D in complex with sotorasib and MRTX1133 respectively, with very high resolution. And this is just an example of our capability to obtain high-quality structure. I personally have never seen a project that has not been positively impacted from the elucidation of the binding mode of the molecules with their targets. Therefore, it is very important that we have this capability in-house to support our projects. One of the greatest innovations in the recent years is the development of small molecules that do not inhibit, but instead induce the degradation of their targets. And these molecules work by inducing the formation of a ternary complex between the molecule itself, the protein target, and an E3 ligase that will ultimately drive the degradation of the protein target thanks to this induced proximity. And this modality is particularly important because it allows to target proteins that are cancer drivers, but do not have catalytic functions, for example. And there are several other potential advantages, like the ability to target different binding sites, to overcome high protein levels, to show prolonged pharmacodynamics effects, to achieve tissue selectivity based on E3 ligase expression, etc. And I will not go into detail in the differences between bifunctional degraders and molecular glues. But one important difference is that while products can be designed rationally, the discovery of molecular glues has been until now more serendipitous. And this is because molecular glues can be very potent in inducing the ternary complex formation, while being weak or very weak binders of the two proteins. And as you can imagine, it's very difficult to screen for such compounds. And actually, there is a lack of adequate hit identification approaches. We have recently presented the results of a screening that was aimed at identifying molecular glues able to induce the formation of a complex between the kinase WEE1, a very well-known and pursued target in oncology, and the E3 ligase cereblon. And to do this screening, we used an innovative technology, spectral shift, that was miniaturized to be able to screen thousands of compounds in a very short time and with very low protein consumption. And we screened in parallel for three conditions: to identify binders of WEE1, binders of cereblon, and binders capable of inducing the formation of the ternary complex. And as I was mentioning before, molecular glues tend to be weak or very weak binders of the two proteins, and therefore they would not possibly be identified with the first two conditions because they work specifically to induce the complex formation. And as you can see from the Venn diagram on the right-hand side of the slide, the screening actually identified some hits in all three conditions, and the molecular glues hits were successfully reconfirmed in follow-up studies. And here is an example. You can see that these molecules, you can see on the left-hand side of the slide that this molecule does not bind WEE1, while it's quite a potent binder of cereblon. But more importantly, if you look at the graph in the middle of the slide, this compound is able to actually potently induce the formation of the ternary complex with a KD of around 340 nanomolar and a very high cooperativity factor. And additionally, we confirmed that this molecule is actually able to induce the degradation of WEE1 in a cellular context, as you can see on the right-hand side of the slide. And so further characterization is ongoing on this and other hits, but these results are a proof of concept that we now have the possibility to directly screen for molecules capable of inducing ternary complex formation. Another modality that has revolutionized the world of oncology and biologics is represented by antibody-drug conjugates. ADCs combine the specificity of monoclonal antibodies with the potency of highly cytotoxic drugs, allowing the selective delivery of the cytotoxic drug to the cancer cells and consequently minimizing systemic toxicity and improving the therapeutic window. And currently, I think there are at least 14 ADCs that have been approved by the FDA and several others that are being tested clinically for treatment of different cancers. Generating an ADC is a very complex process that needs a multidisciplinary approach, the integration of different technologies and capabilities, and the collaboration of teams with different expertise. We have built and are building this expertise that starts from the chemistry, the synthesis and handling of the payloads, the conjugation of the payloads with different linkers using different chemistry approaches to generate the drug linker, the conjugation of the drug linker to the antibody, and the follow-up characterization of the resulting ADC in terms of drug-antibody ratio determination, aggregation, stability, antigen binding, cellular binding and internalization, up to the final anti-cancer effect. And it's very exciting that also this modality is now available for our projects. And I will now leave the word to Alistair that will talk about the importance of identifying biomarkers of response and understanding the drug's mechanism of action to inform clinical development and prevent unforeseen toxicities.
Thank you, Michele. So as Michele said, now we're going to look a little bit more at the cell biology aspect of oncology drug discovery. And what we'll do is we'll start with looking at the ways by which we can identify predictive genomic biomarkers for both sensitivity and resistance. And this is very important because the use of clinical biomarkers as predictors of sensitivity has really transformed the way in which oncology drug discovery is conducted. Initially, tumors were taken and it was more of a shotgun approach. But really, Genentech pioneered the way there with trastuzumab and the clinical trial of that antibody by selecting breast cancer patients specifically for that trial. Obviously, HER2 overexpression being a predominant feature in many breast cancers. Loxo Oncology then took that further more recently with larotrectinib by being agnostic with respect to the tumor type being considered, as long as they had an NTRK fusion. And obviously that related to the target of larotrectinib. And what we saw then was from the advances made by Genentech that there was an improvement in the overall response rate, and then that was taken even further with Loxo's approach with larotrectinib. So how can we go about identifying predictive genomic biomarkers for sensitivity? One way of doing this, and a way that we employ using the Eurofins Discovery OncoPanel platform, is to evaluate a drug against a broad selection, a diverse selection of different human tumor cell lines, especially where we know the genomic composition of those cell lines. So in this particular example, using vemurafenib as a case study, in the graph at the top left, you can see that different tumor types generally respond differently in different ways. Vemurafenib was developed as a small molecule inhibitor of activated BRAF kinase, the advent of which stemmed from the identification of activating mutations in BRAF. And those mutations are seen in a variety of tissues, but notably prevalent among melanomas. And what you can see from the chart here is that melanoma cell lines tend to show more of a sensitivity to vemurafenib as compared with other cell lines, of course, where some of those other cell lines don't contain that genomic driver. So if we look at the drug response of a broad selection of cell lines, and then we look at the genomics, and we identify a resistant and a sensitive population, we can statistically interrogate in each of those populations which genomic drivers correlate with those populations. What's shown on the right, for example, is looking at mutations. And again, for vemurafenib, it's no great surprise that we identify BRAF mutation or amplification of function as correlating strongly with sensitivity. Conversely, we see things like KRAS mutation, P53 mutation, correlating with resistance. And again, this makes sense in context of how vemurafenib works. So we can see here that we can identify good predictive genomic biomarkers for sensitivity of a drug going forward. Now, also looking at ADCs, one of the new therapeutic approaches which is really gaining traction by using the targeted selective ability of an antibody to deliver what would otherwise be a very broad spectrum cytotoxic agent. This is summarized in this particular slide. So in the top left, you can see the waterfall plot generated by looking at the effect of deruxtecan over a range of tumor cell lines, as I'd mentioned on the previous slide. This shows a typical cytotoxic plot where there are some cell lines that are more sensitive than others, but across the collection, most of them show some sensitivity. In the bottom left, you see the similar profile for trastuzumab alone. And in this particular case, you see a very narrow selection of cell lines that respond to this drug, and in many cases, these are driven by the presence of overexpressed HER2, as we would expect. So when trastuzumab is linked to deruxtecan as an ADC, in the plot on the right, what we see are two things. One, we maintain the relative selectivity afforded by trastuzumab. And so even though deruxtecan is part of that therapeutic, you don't see the broad spectrum cytotoxic activity. And this is because of the targeting using trastuzumab. What is interesting is that the selection of cell lines that are sensitive to trastuzumab deruxtecan is slightly enlarged as compared with that seen with trastuzumab alone. Now, another advantage, of course, of something like trastuzumab deruxtecan is that it provides the ability to kill tumor cells with the therapeutic alone. Trastuzumab, in many cases, will show a partial response from the perspective of proliferation, and it also employs ADCC, which is not shown here, but contributes to its therapeutic activity. However, with trastuzumab deruxtecan, you see a cytotoxic response in the cells. And just in the bottom right of the figure on the right-hand side of this particular slide, I'm just showing there the effect of trastuzumab deruxtecan in blue and trastuzumab MMAE, another ADC, in red. And you can see their more cytotoxic responses as compared with trastuzumab alone in the green. So thinking now a little bit more about other cellular aspects of oncology drug discovery. We've talked about some of the new therapeutic modalities and some of the ways in which they work and in which we can identify biomarkers suitable for monitoring as indicators of sensitivity. But what about the effect of drugs in a broader biological consequence? Obviously, this is something that needs to be considered in oncology drug discovery and historically has been a problem with widespread use of harsh chemotherapeutic agents with broad toxicity. What I'm showing here is a summary, an overview of the Biomap platform, which is a human-based phenotypic platform that makes extensive use of normal human primary cell types, often in co-culture. This means that the models that we have, the models, each one looking at a different aspect of human tissue biology or disease, they are using normal primary cell types because they haven't effectively forgotten where they come from. They still behave very much as the way they did in the body. We also have models that cover a variety of different tissues, so there's a good spectrum of different tissue scenarios represented. And the models that we've put together not only employ co-cultures in many cases, but all of the biomarkers that we measure as a means of looking at response of those models to test agents are clinically relevant biomarkers. What makes this platform even more powerful is that we have tested a vast number of different agents against the platform. This includes clinically approved drugs, entities that have been into clinical trials, drugs that have failed because sometimes they can yield very valuable information, small molecules, biologics, peptides, natural products, etc. So we've collectively tested over 4,500 agents. And as such, we have a huge repository of different molecular phenotypic fingerprints that we can generate by testing across 12 different systems as part of a standard panel. Using advanced analytical tools, then, we can look at a new test agent and see how its profile corresponds with what we have in the database. For example, we can identify mechanisms of action for drugs that have been identified using phenotypic drug discovery. This is, in fact, a paradigm that has waxed and waned throughout oncology drug discovery and currently seems to be on the rise again. We can also look for potential toxicity signatures, and these have been identified by looking at different drugs and triangulating biomarkers common to their toxicities. We'll talk a bit more about that a little later. So coming back to trastuzumab deruxtecan and comparison with trastuzumab, this is an evaluation of those two drugs against a broader range of biological models in the Biomap system. And the 12 models here represent different aspects of tissue biology and disease that are not directly related to cancer. So by profiling against this, we can get an idea of the effect of these drugs in other tissue types. The key thing to note here in this particular case, when we are looking at the changes in 148 different protein biomarkers across the 12 models, you can see that in the top, looking at trastuzumab, there is a profile. There are certain changes that we see. Those are reflected by the red, orange, yellow, and green traces that come outside of the gray pattern in the middle, which represents the 95% confidence interval for the vehicle control. So anything outside of that is a meaningful change. What you will also notice is in the 3C system at the left and the HDF3CGF system towards the right-hand side, a wide gray arrow on the x-axis. That indicates systems in which we see a small amount of anti-proliferative activity. Now, when we look at the profile for trastuzumab at the top, and we compare it with that of trastuzumab deruxtecan at the bottom, we see the profiles are very similar. There is shared anti-proliferative activity in the 3C system, largely reflecting decreased proliferation of vascular endothelial cells. And there are other markers that share similar profiles. However, there are a few small differences. In particular, anti-proliferative activity seen in the SAG system and the BT system, which look specifically at T-cell activation and T-cell-mediated B-cell activation, respectively. This illustrates some of the fundamental differences between trastuzumab deruxtecan and trastuzumab alone, but I think you can see that there isn't a dramatically different profile here. And this in part can explain why trastuzumab deruxtecan has a relatively good safety profile as compared with something like trastuzumab, which is also very safe. So let us now take a quick look at immuno-oncology agents and how we might be able to identify those for small molecules. What is shown here is another biomap plot, in this particular case using models of the human tumor microenvironment, in this particular case by co-culturing a human tumor cell line along with normal cell types, whether they be PBMCs, fibroblasts, or human umbilical vein endothelial cells. So what we can see here in the panel is the activity of checkpoint inhibitors when administered. What they show is the ability to restore immune response as compared with the vehicle-treated baseline shown in gray. This is in fact how these drugs work. They do not work by activating the immune system. They simply work by restoring immune cell function, and thus the tumor is identified by the immune system, and then that leads to its destruction. Now, interestingly, if we look at a number of other drugs in these same models, in this particular case on the right-hand side, looking at erlotinib, gemcitabine, and paclitaxel, what you see here is a very interesting restoration of immune response in these same models that, while not quite the same, does in many cases mimic some of the activity seen by the checkpoint inhibitors. So what this means then is that we can also identify immuno-oncology properties of small molecule drugs, and in this particular case, drugs that weren't really even designed to have immuno-oncology properties. In fact, they were in use long before immuno-oncology was really recognized as a transforming way of treating cancers. So in addition to ways of looking at monitoring efficacy and associated biomarkers, and even extending the applicability of small molecules into the immuno-oncology world, another challenging facet of oncology drug discovery, as Michele mentioned at the outset, is attrition. Now, attrition in a large number of cases can often stem from the occurrence of unforeseen safety issues, either in preclinical development or in phase 1 trials. And as you can see here from the figures on this slide, there is still an unacceptably high failure rate. So really the question that emerges here is, are there better in vivo strategies that we can use to try and mitigate this risk and try and reduce the failure rate, reducing attrition and improving clinical success? And if we can do that before clinical evaluation, that results in the moving forward of better drugs that are safer into clinical evaluation. So what I'm showing here is a slide that summarizes standard safety pharmacology assay panel evaluation of two particular drugs, two PI3 kinase inhibitors, umbralisib and idelalisib. These have been long the gold standard for safety evaluation in the drug discovery industry, not just for oncology drug discovery, but really for all therapeutic areas. And they remain so. These assays are perfectly robust, and they are very valuable in identifying activity, especially for small molecules, early in drug discovery, such that use of medicinal chemistry can remove unwanted properties from their profile. These assays are very targeted. They look at a number of targets that are known to have safety issues. And what you can see here in the top is a heat map showing the different profiles of idelalisib and umbralisib. And then when one looks at the significant hits from both, you can see that specifically the IC50 values that are generated for each against the relevant targets. What you can see here is that idelalisib generally shows a slightly safer profile than umbralisib. And when we look at these different activities, we can also plot these to get an idea of potential toxicities, both in terms of a radar plot shown there in the middle, as well as potential organ liability assessment shown in the bottom right. So again, these are targeted assays, and they provide a very good way of identifying quickly, easily, and cost-effectively liabilities that can be very quickly addressed. But what else can we do? I'm going to take a step back now to the Biomap platform that we introduced earlier. I had mentioned that we can use the evaluation across a broad range of models, or systems as we call them, that look at a wide variety of different aspects of normal human tissue biology and even other disease states. Now, I'd mentioned that we profiled a large number of agents in that platform, and if we look at different drugs, different approved drugs that have known toxicities, with that huge database that we have, we've been able to triangulate on different biomarkers for different toxicities. And so what we have here are 9 different toxicity signatures that have been identified, each one associated with a number of drugs that are known to cause that toxicity, as well as pathways and biomarkers that are relevant to that particular toxicity. So what we can do now then is, with a new agent, we can test it in Biomap, and we can look at that molecular phenotypic fingerprint that is generated, and we can ask, does that new agent have any of those hallmarks? Can we identify any of those toxicity signatures? And if this is done early enough, this can help mitigate the risk of seeing this either in preclinical development or even worse, in clinical trials. So using idelalisib as a case study here, this is an approved PI3 kinase delta inhibitor. You can see the Biomap profile at the top. And again, when considering biomarker changes in relation to the 95% confidence interval for the vehicle control, shown in gray, you can see that in many of the systems, there really isn't any applicable activity. This shows a very clean profile. What is particularly notable is some quite significant activity in the BT system. And again, this is no great surprise because of the B-cell receptor activation and its involvement with the PI3 kinase pathway makes absolute sense when one is applying a PI3 kinase inhibitor to those cells. What you will see along the x-axis is the gray arrow indicating some degree of anti-proliferative activity in the BT system, and also in the HDF3 CGF system towards the right. Now, if we take this profile and we use the analytical algorithms that we have to identify whether it contains any toxicity signatures, perhaps unsurprisingly, what we see is really only one toxicity, and that is immunosuppression. And we see that at all 4 doses. And this is a reflection of that immunosuppression we see largely in the BT system there. Of course, this correlates very nicely with what one sees clinically with idelalisib. And so much so that the observed colon and lung inflammations that sometimes occur may result from increased infection. And this probably is related to subsequent inflammation that arises because of that initial suppression of the ability to identify and remove pathologic microbes from the body. Unfortunately, this can also then subsequently lead to potential intestinal perforation, and that is in fact a box warning for idelalisib. But overall, these effects are generally well tolerated, and idelalisib remains an inhibitor, a drug that is approved for a large number of cancers. If we now look at umbralisib in comparison, what we see again, umbralisib being a PI3 kinase delta inhibitor with additional casein kinase 1 epsilon activity, we see that significant response in the BT system there. Again, this reflects the PI3 kinase inhibitory activity of umbralisib. But what you'll also notice, and we've looked at a wider range of concentrations, both going higher and lower, is you see activity in a number of other systems. And interestingly, some of the activities you see there relate to different aspects of human biology tissue remodeling. For example, biomarkers such as alpha smooth muscle actin, various collagens, matrix metalloproteases, and things like that. So clearly this drug has a very slightly different profile as compared with idelalisib. And in fact, when we do the toxicity signature analysis, again, it's no great surprise that we see an inhibition of immune response or immunosuppression. But what we also see are a few other toxicities, including skin irritation, organ toxicity, and skin sensitization, as well as elements of vascular toxicity. What you will also notice that potentially relate to some of these toxicities are the gray arrows along the x-axis. Rather than seeing anti-proliferative activity just in the BT system, we also see it in the 3C, the SAG, the CAS-M3C, and the HDF3-CGF systems. And what you will also notice, in fact, is in the BT system, the small black arrow that indicates in that particular system where we do also see elements of cytotoxicity. So it's easy to see then why in this particular case there's a slightly more toxic profile of umbralisib. And although initially approved as a therapeutic for oncology, it was subsequently withdrawn due to long-term side effects and overall a lack of therapeutic benefit versus the increased risk of death from taking this drug. If we think about some of the biomarkers here, many of them, as I say, make absolute sense in context of a PI3 kinase inhibitor. But if you look at some of those tissue remodeling biomarkers, one raises the intriguing question, is this potentially a way of identifying proactively drugs that might subsequently be withdrawn because of tissue remodeling? The short space of clinical trials doesn't always allow us to evaluate that. Whereas once a drug is approved and is being prescribed and taken for long periods of time, only then will those longer-term effects come into play. So again, this raises the interesting possibility of using the Biomap platform not only to mitigate immediately obvious risks and toxicity, but also potentially to question whether there might be longer-term risks using this toxicity signature analysis. In his earlier slides, Michele had also mentioned some of the challenges with drug discovery, and including failures not only in phase 1 trials, but also in phase 2 trials, clinical efficacy. Just shown here is a very quick case study looking at sorafenib against vemurafenib. Sorafenib was initially developed potentially as a BRAF kinase inhibitor following the discovery of BRAF mutations associated with various cancers. If you look in the top left, this shows, again, another waterfall plot using the OncoPanel platform. And we're looking at the sensitive cell line shown in the gray box on the left-hand side. And what you will notice is we've highlighted there the cell lines that bear the BRAF V600E mutation with red lines. And unfortunately, you can see here that activity of sorafenib doesn't really correlate with the presence of BRAF mutation. Just for reference, there are a few other cell lines in there that are shown, in particular those with yellow, that represent cell lines with c-KIT overexpression, BRAF being a potential preferred effector for c-KIT, and also NRAS mutation. It's hard to see, but there are a couple of cell lines in there with a black coloration. But again, none of these really correlate with sorafenib activity. If you look in the bottom left, in contrast, vemurafenib, a BRAF-targeted agent, you see a very strong correlation there with sensitive cell lines and the presence of the BRAF-activating mutation. Furthermore, cell lines that bear c-KIT overexpression or NRAS mutation, constitutively active mutation, they all tend to cluster towards that sensitive side of the waterfall plot. And again, it's no great surprise that vemurafenib, of course, was approved as a therapeutic for BRAF-driven cancers. So what about sorafenib then? Obviously, with hindsight, we know that sorafenib has other activities, other kinase activities. And in fact, it is this which has led to its approval for a variety of cancers that are not intrinsically driven by BRAF mutation, in particular those such as hepatocellular carcinoma and renal cell carcinoma. And that's actually reflected by the xenograft data shown on the right of this slide. At the bottom there in blue, you can see activity of sorafenib, which is a very impressive activity against, in this particular case, the Renker model. This is a mouse kidney model, as compared, interestingly, with several different immune checkpoint inhibitors. These are shown in the yellow, green, purple, and orange traces above, the orange being naive treatment. So in this particular case, we can actually show why two different drugs that were ostensibly developed for the same ultimate aim ended up with different therapeutic uses. And in fact, in certain cases, we can look at repurposing of drugs with a combination of things like the OncoPanel platform here, the Biomap platform, and of course by interrogation of drugs in an in vivo scenario. So I think you've seen from the slides that we've presented throughout this webinar, there are various aspects to oncology drug discovery that initially present challenges, but there are ways that we can overcome those challenges. Not only from the therapeutic perspective, looking at different modalities for drug discovery and solutions that can help evaluate those drugs in early discovery and early preclinical development, but also ways in which we can evaluate other facets of oncology drugs, including things such as safety. Eurofins Discovery offers a broad range of oncology services such that we can provide end-to-end assistance with all of your programs. This ranges the whole gamut from chemistry resources right the way through to different in vitro assays, safety pharmacology assays, cell biology assays, and of course properties such as ADME and in vivo activity. And in fact, we can also offer these for companies that are working in a virtual environment as integrated drug discovery programs where we provide full program management. So just in summary here, I'd like to wrap up and show you with the slides that you've seen today and the presentation that Michaela and I have given, ways in which Eurofins Discovery has addressed some of the ongoing and some of the new challenges in oncology drug discovery. And that's reflected in the more than 23,000 different solutions that we provide for you to help kickstart your programs. We've talked about new target types that have emerged, such as helicases and KRAS, and some of the challenges inherent with those targets, and how we have provided solutions to help you develop drugs in those areas. I mentioned briefly on the last slide that we can also provide integrated approaches. There are obviously a number of companies that are virtual in nature, so we can provide those end-to-end solutions along with program management. Simply put, we can provide solutions really at any stage of your drug discovery needs. Obviously, with precision medicine focus and the translational solutions we have, and we've given some examples using OncoPanel and the Biomap platform, as well as custom in vitro and ex vivo biomarker measurement services, we can also employ those alongside some of those traditional solutions. We talked about safety pharmacology and some of the standard sort of approaches. We can bring those all together to provide a powerful solution for pretty much any drug discovery program. And as with the oncology field, there are new developments, new target types that come into being, and we evolve with that. We continuously expand our capabilities to meet those needs. So let us know about your needs. We are always happy to consider what you may have, what you may need, and how we can address those needs. If you would like to discuss any aspects of this webinar or particular needs you have with us, please feel free to reach out at any moment. Please feel free to contact either myself or Michele at the email addresses shown. We also have a QR code associated with that. We also have a number of assets, brochures, flyers, white papers, etc. Two of them are just shown here. And again, there's a QR code for accessing that. And you can also review our services on our website, both the Eurofins Discovery website, and we also offer a full range of products that can be useful in supporting oncology drug discovery through Eurofins DiscoverX. And the websites are shown for both of those, along with the QR code to access. So Michele and I would like to thank you all very much for your time and attention throughout this webinar. We'd also like to thank the organizers for the opportunity to present, and we would be more than happy to answer any questions you might have now in the Q&A session. Thank you.
Thank you so much, Michele and Alistair, for that excellent presentation. It was chock full of great information for our audience. And I just want to remind the audience that you can ask questions through the Q&A box on your screen. And now we will open it up for questions. This audience member, I believe, is looking, has a question for Michele. They want to know the benefits, drawbacks, and differences of drug discovery via manual approach versus drug discovery using machine learning.
Yeah, thanks, Melissa. And actually, that's a great question. Not easy to answer in a very short time, but let's say that I see AI and machine learning as ways to, let's say, handle and extract insights from the large volume and complex data that we now have. So let's say we have roughly two ways AI can help. One is in target discovery to integrate all the multi-omic data sets to identify novel cancer vulnerabilities and identify synthetic lethal interactions. And on the other hand, on the hit optimization and lead optimization part, AI-driven molecule design is used to design compounds with optimal properties, predict ADME profiles, and suggest modifications to reduce off-target toxicity, improve selectivity, and so on. So I would say that these tools are very important because they will allow to accelerate the decision-making process during the project and the testing of the hypothesis. So probably helping us move from let's say, a trial and error to a more predictive science. And obviously, the key point, however, is to feed these computational models, these AI models, with high-quality data sets, and then, therefore, integrating these tools into the experimental workflows.
Okay, thank you. Michele, this question is also for you. This audience member is asking, where do you see the greatest opportunity to improve preclinical to clinical translation in oncology?
Okay, yeah. Also, this is a very nice question. So I probably mentioned during the presentation, but probably did not really elaborate on this. One opportunity is improving the translational relevance of the preclinical models. So in oncology drug discovery, we usually rely on models which are the in vitro cell lines and derived from tumors and in vitro xenograft that probably do not recapitulate exactly the human tumor biology, especially for what regards to the heterogeneity, both at the intratumoral level and also the heterogeneity that comes from the patient heterogeneity, the fact that each patient has a different genetic background, for example. So there is a trend of incorporating what are believed to be more predictive models, like organoids, co-culture system, patient-derived xenograft, that could allow to evaluate better the efficacy in a context that is closer to the clinical scenarios. And as I explained also during the talk, I believe firmly in improving the mechanistic understanding of how the drug works, what pathways it modulates, what are the context-dependent effects, if it has synthetic lethal interactions. This will allow us to understand, for example, which patient population is most likely to respond. And also, I think in oncology drug discovery, with respect to other therapeutic areas, there is we are less used at integrating biomarker strategies from early on, whether for target engagement, pharmacodynamics, or patient selection. I think this is something that is very important that will translate in a better clinical success rate.
Okay, thank you very much. This audience member is asking the questions for Alistair. What are the ways to reduce the detrimental effect of the drug? Actually, these are increasing day by day.
Yeah, that's a great question. And as we demonstrated in the seminar, there are still a lot of effects that are seen in clinical development, in particular in phase 1 trials. So a key really is to try to identify not only what those effects are, but ideally what the causal agents are for those effects. For example, the safety targets and things like that earlier on. If that's done earlier on, especially for small molecule drug discovery programs, medicinal chemistry can then be employed to dial that undesirable quality out of the molecule. And for example, the Biomap platform that we spoke about during the webinar, that's actually a very good tool for identifying some of those more arcane toxicity events or causes of toxicities that may not be immediately apparent from conventional assays. So conducting studies like that to really identify if there are any liabilities earlier, that allows things to be done before you get to the stages of preclinical development and ultimately clinical trials. And of course, that helps improve the overall success of the molecule going forward.
Okay, thank you very much for that excellent answer. We also have another question for Michele. What are the criteria to decide whether a novel target is worth pursuing, especially when clinical validation is limited or absent?
That's also a great question. I would say there are three large guiding principles. One is that we are looking for a strong biological rationale. So ideally, you have multiple lines of evidence converging on the target's role in tumor survival or maintenance. And evidence coming from genomics, transcriptomics, functional screen like CRISPR or RNA interference, clinical correlation with poor prognosis, etc. The second is the target tractability. Can we modulate the target with a modality that is feasible, either a small molecule, an antibody, a degraders, etc. And the third one is the technical feasibility. Can we build the right assay? Do we have the relevant disease models? So it's really about making together these three parts: biological relevance, tractability, and technical feasibility, that all together make to take the decision whether a target is valid or not.
Okay, thank you very much. And we have time for just one more question. We're going to sneak this in. This one is for Alistair. This audience member is asking, can you share examples of when early understanding of a molecule's mechanism of action changed the course of a discovery project? Great question.
Yes, that is something that does occur from time to time. Once a target is selected, there may be either a change in how that target is modulated or the properties of the end resulting drug that need to change as a result of chemical tractability and things like that. A particular example of that would be for the oncology space, if one is developing an inhibitor towards a particular target and one sees cytostatic activity, ideally one would want to go into a clinical trial with something that is tumoricidal, so cytotoxic towards tumor cells, but not normal cells. But in the event, either the nature of the target or just simply that series really only allows cytostatic behavior, that has an impact on the PK and the way in which that drug is dosed. For example, if a drug is cytostatic and needs to be taken repeatedly, then something like an IV administration is not going to be a viable way forward. Whereas if it is oral, then that is a lot more attractive from a quality of life perspective. Of course, if a target or the drug that's designed towards that target is tumoricidal, then IV is acceptable there. Although, of course, oral is preferred, but IV is acceptable in that case. And we still use a lot of drugs that are administered by IV because the idea is that once the tumor is gone and dead and hopefully completely cleared, then that therapy is not needed any further, as opposed to something that is cytostatic, where you might be needing to take that drug for a much more protracted period of time.
Thank you so much, Alistair. And that's about all the time that we have for today. Thank you again to Michele Modugno and Dr. Alistair King for that excellent presentation. Any questions we didn't get to during the webinar will be answered by email. Thank you to the participants for being such a great audience today, and On24 for the technology and production services. A big thank you to Eurofins Discovery for the sponsorship that made this interactive webcast possible. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. And for CNN webinars, I'm Melissa O'Mara. Goodbye.